So is it, are you here? Yes. Is it better now? Kindly let me know. The clarity is now okay. Everything is okay now. Now it's fine. Okay, good. So I have to do it again because it's not okay. Okay, everything is fine. Okay, good. So a very good evening to all of you. How are you all? And how is your day today? Everything is fine. Today is Monday. So let me know how is your day today. Everything is fine. Yes. Good evening to all of you. We have Juveria here. Good evening, Gayatri, Anushri, Shubham. Not Shubham. It's Shubh Mahatre. Then we have Vidnya, Namde, Lekha, Vedika and Ankita. So good evening to all of you. Just waiting for another students to join. I hope they will get a new link also. Sakshi is also here. So good evening to all of you. Pavitra is also here. They all are good. She is saying all good, ma'am. Okay, very good. Devita is here. Good evening, Devita. So how are you all? I hope you all will be fine, definitely. And how is your day today? Today is Monday. Maybe you have lots of assignments for your college also, or some type of test maybe also. I don't know. So let me know what is your activity today. What is the type of activity you have done today? Let me know. Yes, Ganesh, good evening. A very good evening to you also. So we have number of students who are joining. Jeet is also here. Yes, Prachi, good evening. Jeet, now is it okay? Everything is fine now? Because first the webcam image is too big. Now is it everything is fine now? Kindly let me know. Yes, Jeet is also here. Okay, good. So everything is fine in your side. Today only lecture. Okay. So today is a busy day for all of you also because you have a lecture. It's a good thing. Okay, your practicals got cancelled. In our university we have a practical which we have to conduct in the month of November. So we have to do it. We have practicals in the month of November, maybe from 15th of November. So you are lucky your practicals got cancelled. Maybe from your theory papers or theory exams, they will give you the assessment or the marks on the basis of your theory work, or maybe they will conduct your viva. Maybe this may be the option also, because like in our university we are doing online or video calling. We are taking their viva also, so we have to do these type of duties also. So what about your theory exam? Tomorrow we have practical. Okay. Yes, Ankita, I am also fine. Today I just went to the university and I also have lots of work to do. I am just asking that what about your final theory exams? When it will be? It will be in the month of December or in November? When will you have your final exams? Okay, only for today. Okay, so you have your practicals within these coming days. Okay, good. So when will be your final exams? In the month of November or in the month of December? When will be your final exams? Okay. So Prachi is saying That regular practicals. Okay. Okay, good. Okay, they are planning internal exams like MST, basically mid semester examination before Diwali. Okay. So yes, maybe your exams and your final exams maybe in the last of November or in the first week of December, maybe, because these are the criteria which is there in our university. They are pursuing this type of things. Like we have the MST nowadays, then in the December or in the month of November we have their final exams in the last week of November, basically. Finals not yet decided. Okay, 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 good. And today you all have your regular classes also. You have your regular classes, the theory classes today. Okay, if everything, Devashish is saying, if everything is taken normally, then they must be before Diwali. Okay, good, good. Yes, now there is a Dussehra within this. I think on 25th, maybe on Sunday we have a Dussehra, and after 20 days of it we have a Diwali. So nowadays or in this year, we are not, we are not that much aware of the festivals, or we are not enjoying the festivals as we were doing in the previous years. So we are not like Navratri is there in I think from yesterday we have a Navratri, first Navratri. So the events are there. First we have Ganesh Chaturthi. I think in Mumbai you all have Ganesh Ji in your home, definitely, because it is a celebration or it's a function of or a festival of Mumbai. So you must have those types of activities also. What will be the last day of MBBT? What will be the last day of MBBT? So it's up to you how long you want to take the classes. I'm here if you want to take it for 22. I'm here if you want. It's up to you, because according to me or my priority is up to Wednesday. So Wednesday is we have on 21st. So if you want to continue it for 22nd or something like that, if you have something in your mind or you want to discuss something, then most welcome. So I am here, so there is not any worry. You can tell me how long you want to take this session, because I am always there, no issues at all. So basically I think it is up to Wednesday. Then if some topics are there which left, or some issues are there, some queries are there, then please let me know. We will extend it further. There is not any problem in it. So it's up to you only. You have your class. Until you complete the portion, you take the lectures. Okay. Okay, yes. Oh, I will take definitely. And even if some students or any one of you, you want to, some topics or queries are there, we will take special classes also. There is no any issue at all. Okay, Devashish. I will try my best to complete the topics within these 2 or 3 days. I will try, because only 2 topics are left now. The important ones I have already covered before. So like we have molecular biology techniques, which is PCR and ELISA, then the application of molecular biology techniques, then we have recombinant DNA technology and its application. So these things will be covered today. Flow cytometry we have done, separation of molecules we have done, protein quantification, separation and purification we have done. So recombinant technology, there is a 2 part, part 1 and part 2. I will cover it in a single part. So if anything another is left, kindly let me know, because the syllabus which I have is only that much. If anything other is there, kindly let me know. So it is up to you how Jeet is here. Jeet, kindly consult with or talk with the batchmates or your classmates and let me know only. Similarly, Juveria, you also talk with your friends or your batchmates and let me know. So there is no issue at all. Even individual person can talk with me, no issues at all. So I just want to say you one more thing, that in the yesterday session, which is for 3 hours, we have only few students who attend the session. Like I seen the video today also when I was in department. So I found that there are only very few students participate in this lecture or especially on Sunday. So I request all of you to go through this video once again. Are you getting my points? You all are not present in the lecture series. Even it's a holiday, and on Sunday, that is on yesterday, I covered lots of parts. So even we take a break for 20 or 25 minutes in between, but I found that all students are not present in this particular lectures which we take on Sunday. So that's my request to all of you, that kindly go through the video. Generally we have 200 views on a single video, that means that much persons are watching it, but on the yesterday video we have only 80 views. So the only few students are there who are attending the session. So it's my request to all of you that kindly go through it once again. So if you have any other query, kindly let me know, please. So you don't need to worry about the this, that your syllabus will not be complete. It is almost completed, but if you have some type of query, if you want to know something another or any another technique in MBBT, kindly let me know. You have to share the things with me, then I will able to get the things. And even you are my first batch, so I have a special attachment with all of you, and I want that you will remain in touch with me also. So there is no issue of doing the class more than the 10 days or something like that. There is no issue from my side. Yes, it is the thing that if you are busy, if you have your own work or own schedule, then I will not force you at all, but you have to let me know also. So by your replying, I will able to get your point. So Prati is here, she is saying definitely ma'am, yes. So you have to tell me that yes, this is also here. So you are mature students, you can talk with each other and you can share your point of view with me also. So there is not any type of problem in it. So if there is any another query, then let me know. Otherwise we will move to our today's lecture, which is basically on ELISA and recombinant DNA technology. The topic recombinant DNA technology, why I taken it late, because recombinant DNA technology, you all know about it. This is a technique or technology you must be aware of. Even you have read it in your theory classes also. That is why this topic I have taken late. No, it's not buffering, Ankita. Today it's fine. The signal is also good from my side. So you just switch off your internet once, and then I think you will get the signal. Ankita, kindly switch off your net first, because today I am finding a good signal from my side also. So shall we start now? Yes, I am just going to start. So it's my humble request to all of you that kindly go through the yesterday videos which we have taken for 3-hour long session, because I think lots of students are not present there. Even by your messages I am able to know you, I am able to remember your names also. So I am fine that lots of students are not there even on Sunday, but it's okay, sometimes you have a work. So it's my request to all of you that kindly go through yesterday's video. Okay. So Floris is also here, Saurabh is here, and then we have Ethel is also here. So a very good evening to all of you. Now just start the video. Yes, good. Yes, Mitali is also here. So if you have any type of other query or something like that, kindly let me know. So we will start with ELISA first. Even I have taken some videos, I have also taken some videos for ELISA and recombinant DNA technology. Yes. So now we will start. And yes, one more thing I want to say to all of you, that in the night or after your session, we have a class from 8 to 10 of biochemistry. So if you subscribe my channel, you will definitely get a notification also. So if you want to learn something about biochemistry or something another things instead of MBBT, so you are most welcome. You can join me on the live session also. Even you can message me there also, so that I came to know that you are my MBBT students. So you are most welcome for the biochemistry classes also. So there is not any issue that you are only for MBBT. No, if you want to learn something, then you are most welcome to join the biochemistry session also. Yes, you are there, but I am some students from MBBT message me, but I don't found your message basically. It's okay if you are not enrolled, it's okay, Devita. Just link on or just click on my link, which you must get a notification from that the crash courses is live now. So just open it and just be live with me. There is not any issue regarding it. So it's not that you are enrolled or not enrolled. If you want to learn something, then you are most welcome from my side. Yes. So Devita, if you subscribe my channel Crash Courses, then you definitely get the notification that Crash Courses is live now, and then you have to just click it and you will be there with me. So first of all, subscribe the channel, and the timings are from 8 onwards, 8 p.m. to 10 p.m. So some students are there during our some students are there yesterday from 8 p.m. to onwards. So you are most welcome also. And today is the first lecture basically today. So I have to start with nucleic acids and DNA and RNA. So if you are interested, then you are most welcome. Okay, so now let's start. We are already late. We will start now. So we will start with the technique which is known as ELISA. So ELISA is basically enzyme-linked immunosorbent assay. It is the full form of ELISA is enzyme-linked immunosorbent assay. It is a common laboratory technique which is used to measure the concentration of an analyte in a solution. So basically, the ELISA technique is generally used to know the concentration of antibodies and antigens. So you must aware of antibodies and antigens. So ELISA technique, this technique is specifically used to know the concentration of antibodies and antigens. Antibodies and antigens are the part of immunology. So they play a very important role in our own body. So ELISA or enzyme-linked immunosorbent assay is commonly known as ELISA, where antigen-antibody interaction is monitored by enzyme measurement. So according to the name suggests, it's an enzyme-linked. In this technique, the concentration of antigen or antibody is measured by the enzymes which are linked together. So in a biochem section, I also have an enzyme, the full lecture on enzymes. So it's a similar in principle to radio or immunoassay, but it depends on an enzyme. So our main focus in this technique, that is in ELISA, our main focus is on enzyme rather than a radioactive level. So we have another technique which is radio immunoassay, in which our focus is on radioactive substance or radioactive labeling. But here in the case of ELISA, our main focus is on enzyme, the working of the enzyme, that how this enzyme will helpful for us to know the concentration of antigen or antibody, or you can also say to know the reaction between antigen and antibody. So an enzyme conjugate with an antibody. Enzyme, what will enzyme do? It conjugate or it will bind with an antibody, reacts with a colorless substrate to generate a colored reaction product. Such a substrate is called a chromogenic substrate. So what happened basically, an enzyme which is in conjugation with the antibody, it reacts with a colorless substrate or colorless product which is known as chromogenic substrate to produce some effect. So ELISA use an enzyme to detect the binding of antigen and antibody. So basically it is a principle, or it is the reaction between the antigen and antibody. So we have to know the binding or the interaction between the antigen and an antibody. So enzyme converts a colorless substance which is chromogen to a colored product, indicating the presence of antigen-antibody binding. So in ELISA, our main focus is on antigen-antibody interaction. So our main focus is on antigen-antibody interaction with the help of specific enzyme. So this enzyme is there to know the binding or to know the interaction between antigen-antibody. So in ELISA, we have to know the concentration or we have to know the binding between antigen and antibody, but with the help of a specific enzyme. So ELISA can be used to detect either the presence of antigens or antibodies, depending how the test is designed. A number of enzymes have been employed for ELISA, including the types of enzymes which are used in ELISA, because our main focus is basically the enzymes which will helpful for to know the antigen antibody interaction. So type of enzymes which are used in this technique is alkaline phosphatase. So from this phosphatase you are able to know that they are enzymes. Then we have Horseradish peroxidase. Then we have Galactosidase. So these are the three enzymes which was prominent in ELISA. So these are the three main enzymes. These assays approach the sensitivity of RIAS and have the advantage for being safer and less costly. So as in this radioactive assay or radioactivity technique, we are using the radioactive substance which are quite harmful for ourselves. So this technique in which we are using enzymes, it is quite safe and it is also little bit less costly. So we have to remember the point. It is enzyme-linked immunosorbent assay. Antigen antibody of interest is absorbed on a plastic surface. I will show you the video also, and even in the PPT, that we have a plastic surface that is the part of you can say of the apparatus basically. So the plastic surface is known as sorbent. Here what will happen, antigen and antibody will be absorbed on this sorbent. Now antigen is recognized by specific antibodies. We all know about it, that antigen is recognized by specific antibodies. That is why we have antigen antibody interaction. But this antibody which is specific to the antigen is recognized by a second antibody which has enzyme attached. So what is basically this? That we have an antigen is represented by Ag, which will bind with antibody 1. Okay, we have to study their interaction. But what happens, this antibody 1 will further bind or further recognized by the antibody 2 which have enzyme attached. So antigen is antigen will going to bind with a specific antibody, which is antibody 1, but that antibody 1, first that antibody 1 will bind with antibody 2, which has an enzyme attached. So this is the history that it just starts like radioamino assay, which is little bit harmful for us. It starts in 1960, and then from 1970 onwards we have ELISA to perform. So these are the two persons or the scientists who work on the technique. So this is the thing basically, this is the apparatus. So this plastic surface is known as sorbent, So usually we have a microtiter plate having 8 formats. So here 1, 2, 3, 8. So we have 8 formats on this side and we have 12 on this side. So this is the apparatus basically, and this is a solid phase. So this is made up of plastic or some type of like that surface. Here we have to do antigen antibody interaction. Then number one thing which we have to do is the adsorption on the plastic surface or sorbent basically we are saying. So the process of adding an antigen antibody diluted in buffer, so it attaches to the solid phase. So first of all what we have to do, we have to introduce the antigen antibody in all these wells. Then the another step is washing. The simple flooding or emptying the wells with a buffer solution to separate bound from unbound reagents in ELISA. So there is a adsorption, then we have to do the washing to remove out the excess things or to wash out the wells. Then we have antigen. What is antigen? Any molecule that elicits the production of antibody when introduced into a body. So what will happen? This antigen will bind with the specific antibody. So antibodies are the proteins produced in response to antigenic stimuli. So antibodies, when they will be produced, when we have the antigen stimulus or when the antigens are there to stimulate the antibodies. Then we have enzyme conjugate. The enzyme which is the most important one in ELISA is the enzyme. So we have to remember the name of the enzymes which are there. So an enzyme that is attached irreversibly to an antibody, basically to the antibody 2. So we have 2 antibodies here and a single antigen. Like the example is Horseradish peroxidase. So this is the enzyme. Then we have chromogen. These are only the terms which are there in ELISA basically. So what is a chromogen? A chemical alters color as a result of an enzyme. So what happens when we add horseradish, when we are adding horseradish peroxidase, what will happen? There is a change in a color. A chemical alters color as a result of enzyme interaction with the substrate. So we have a substrate. When we introduce the enzyme, it will change its color. The stopping is what? When we are stopping the reaction. Reading is what? When we able to read the concentration of the color or the concentration basically under the spectrophotometer. So the main thing, the main points are there. We have an antigen here and we have an antibody. First thing, but for this antibody, we have another antibody which is antibody 2, which has specific enzyme. So you can see, then antigen antibody is like a key fits into the lock. But another things are there, enzyme conjugate substrates bound to a secondary antibody that binds with the antigen antibody complex. So we have antibody. Let me show you in the diagram. So we have antibody. If there is some diagram. Yes, let's discuss like this thing. So we have antigen here. And we have a primary antibody which is specific to the antigen. So this is a lock and key mechanism. When we have a specific antigen antibody interaction, so this is a lock and key mechanism that antibody and antigens are attached with each other. Then what happens? We will do the washing to remove the excess things. Then after it we have another step, which is the introduction of the secondary antibody, which has a specific enzyme attached with it. So this is the secondary antibody with a specific enzyme attached with it. Now what will happen when we again wash the excess of the things? When we watch it, so what will happen? It will changes its color. Substrate addition, signal detection and quantification. What will happen? Now if you are adding the substrate here, like this blue one is the substrate here, then there is a changes in the color of the well, particularly the well. So you can see here we have no color, but here when this enzyme binds with the specific substrate, it leads to the changes in the color. So are you getting my point? So this is the basic fundamental of the our ELISA. Like again in this case we have antibodies, or like in this case antibodies are bind here. Now we add antigens which are specific to it. Then we are adding the secondary antibody with a specific enzyme or specific enzyme. Now what will happen? This substrate binds with the enzyme and it will produce some type of color which is directly proportional to the signal strength is directly proportional to the concentration of the analyte. So this is the basic concept, and by these interaction between antigen antibody along with the secondary antibody which has a specific enzyme attached to it is known as ELISA. Let me show you one more diagram. Like here again, what will happen here? Just trying to tell you, we have specific antibody, antigen attached, but we have another antigens also present. You are saying the circular one, but they are extra. They are not fitting into lock and key mechanism. So they will be washed out because they are not lock and key mechanism. They are not binding with the antibody. So when we are doing the signal detection, what will happen? What will happen here? You can see they are also present, and analyte concentration signal strength is inversely proportional to the concentration of the analyte. So these are the types of ELISA. Let me show you one more diagram to clear it to you. So this is what I am basically saying, like this is a plastic or a cell surface which I show you before, the plastic surface or the well surface. So we have an antigen, like we have an antigen on which we bind the specific antibody. On this antibody we have another antibody. Are you getting my point? We have another. In this direct we have only antigen and antibody interaction with the tag only, that is with the enzyme only. But in indirect we have two antibodies. In indirect ELISA, I will show you. So if you are able to understand this diagram, we have a one antibody and we have a suppose we have one antibody and we have an antigen. For this antigen we have another antibody which has a enzyme attached, and then we will introduce the substrate. Like here the substrate is of white color, but when this substrate comes in contact with HRP, that is horseradish peroxidase enzyme, it will change its color. So by checking the concentration of this color, we are able to detect the signals or the result of ELISA. I will also show you here. This is a competitive ELISA. These are types of ELISA, but the basic thing is same here. We have antigens here, we have specific antibody to it. We have antigens here, we have a specific antibody to it. Then we will introduce the secondary antibody which has enzyme binded to it. So we will wash it and you will able to see the change in the color of the solution. These are the types of antibodies. I will discuss it also, but I am just focusing on the basic concept right here. So in this competitive ELISA you have two types of interactions. You have two types of antigen antibody mixture, so the results are something different. I will show you. So like in sandwich ELISA, we have antigen antibody interaction, then we wash, then we add the enzyme. They will form the, here you can see the antigens are in between the two antibodies. Like here the antigens are downward, but here the antibodies are present, this one in between the two. So basically what is it? It is, we have antigens, then we have antibody 1, then we have antibody 2. But in sandwich ELISA what happens? We have antibody 1, then we have antigen, then we have antibody 2. So this is sandwich ELISA. So I will just showing you the ELISA, the basic concepts of ELISA. So let me know whether you get it, then I will move forward. I will tell you again. I hope you get it. So first of all, let's see the equipment. So this is a plastic plate. You are saying it. So if you are saying there is a change in a color, that's there is a reaction between the enzyme and the substrate. So you are saying this is the plastic plate. Then we have micropipette. You all know about it, by which we are able to put the sample. These are washing device. So this is a micropipette washer. So this is another important thing in ELISA, that after every reaction, like first between antigen and antibody 1, and then between antigen antibody complex and antibody 2, you have to wash regularly in between. So washing is very important in the ELISA. Washing is very much important in ELISA to remove the excess attached type of interaction. So these are the reagents which are used in the in the ELISA, like we have buffer, then we have washing buffer, we have blocking buffer, then we have enzymes, which is the most important one, like we have Horseradish peroxidase, and then we must have a chromogenic substrate. There is enzyme substrate reaction. This substrate like trimethyl benzidine, which is TMB, will interact with this horseradish. So there is a change in color when there is an interaction between enzyme and substrate. So these are the only one which are important, that we have an enzyme and we have a substrate. So you must have read I think enzymes also. So enzymes and substrate will make enzyme substrate together, and then there will be a change in color. So with the introduction of enzyme and your substrate, there is a change in color. So color will get changed. So are you getting my point? So there is a change in color. I am not able to write it. So this is enzyme and we have an interaction between the substrate. You have to remember this thing. So this is a general thing, that we have an antigen in a well. Which well? This well. Only one, single one. So we have an antigen in the plate. Then we have to do, like they are doing, blocking buffer is added to block the remaining protein binding site. So antigen we will add, then we have to add first of all we have to add the antigens. Then in the antigen we have to add the buffer. Then we will introduce the antibodies which will bind with the antigens. After we will add another secondary antibody which has an enzyme attached with it. If you are able to see, this blue one is the antibody and this red one is the enzyme. Then we will introduce the substrate which is TMB. What is TMB? Well, trimethyl benzidine. So we introduce TMB. So when this TMB interact with this enzyme, we will get the colored product. So when secondary antibody conjugate is then added, which recognize and binds to the primary antibody. Now this TMB, this substrate, is converted by HRP to detectable form. So the enzyme will convert the substrate into a colored product. This is the basic principle of ELISA. If you are able to understand it, if you are able to understand this, then you are able to understand all the forms of ELISA, either it is sandwich or indirect or competitive. We have 2-3 types of forms of ELISA. So if you can see here again, here what happened, like here what we do here, we have antigen first, but here in this case we have antibody first. So antibody is there. Now what we will do, there is an interaction with the antigens, then we have to wash it again, and I think it's a sandwich ELISA because 2 antibodies are there in between the sample. Not in between the sample, 2 antibodies are there in between the antigen. So the secondary antibody contains our enzyme. The enzyme will attach or will bind with the substrate, and then there is a change in a color. So here the color is blue, but later on it becomes yellow. So there is a change in a color. So that is basically ELISA. So let me check whether you are getting it or I am only one who is speaking. Let me check. Ma'am, in the diagram, what the so all of you are asking about which diagram basically. So I think now you will be able to get it, because we have discussed it very thoroughly and deeply. Now we will move forward to the types. Okay, it's a very easy technique. You have to do nothing. You have antigen will bind with the antibody. That antibody will bind with the, or antigen antibody complex will bind with the secondary antibody. Those secondary antibody has an enzyme attached. That enzyme will bind with the substratum and it leads to change in color. The change in color will be notified under the spectrophotometer. So this is the basic concept. And let me show you the video also, so you will get more clarity. Let me show you one video, but I don't know which video is it. I think this one. Today I download the videos because sometimes there are lots of technology. 1 minute please. So there are lots of ads in between the videos.
Process of recombinant DNA.
So there are lots of ads in between. I think this is
ELISA is used.
So okay, I hope it is visible to all of you. Are you getting it? Is it visible to all? Okay. Yeah. Okay, let's start then.
Hello friends, in this animation I am going to explain about ELISA or enzyme-linked immunosorbent assay. ELISA is used to detect antigens of many diseases like AIDS, tuberculosis, malaria, etc. ELISA is based on the principle of reaction between enzyme and the substrate to detect the antigen present in the sample. So antigen will be taken and the enzyme-linked antibody will be added, which will form a antigen antibody complex. Now we will add specified substrate for that enzyme and gives result in the form of color change due to enzyme substrate reaction. Now coming to the methodology of ELISA. First we will take a polystyrene 96 Wells microtiter plate, then add the sample which contains antigen. The sample may be patient's blood or serum or any other part. So antigen will attach to the surface of the wells of microtiter plate. Now wash the microtiter plate by using tris phosphate buffer of pH 7.4 and 0.01 to 0.05% of twin 20 detergent solution. It will remove unwanted materials from the wells of microtiter plate. After that, add specified enzyme-linked antibody to the wells of the microtiter plate, which will form antigen antibody complex. Now again wash the microtiter plate by using same buffer solution like this. After washing, specified substrate for the enzyme is added and enzyme substrate reaction takes place, which forms blue color in general. Now add acid like H2SO4. So color of this solution in microtiter plate becomes yellow. So at the end, this yellow colored solution will be observed under colorimeter in a specified wavelength and the absorbance is determined, and we can conclude that the ELISA test is positive or negative. Now coming to the different types of ELISA. So there are 4 types of ELISA test is carried out, which are direct ELISA, indirect ELISA, sandwich ELISA and a competitive ELISA. Now coming to the direct ELISA, which is already discussed in the methodology. So first we will take a sample which contains antigen. Then we will add enzyme linked antibody which forms complex with antigen like this. Now add the specified substrate for the enzyme used, and both will react and forms a product which is blue in color like this. Now coming to the second type of ELISA, which is indirect ELISA. So in this type of ELISA we will take the sample and then add antibody which will form a antigen antibody complex. Now we will add an enzyme linked secondary antibody which will react with the primary antibody, and finally specified substrate is added. So substrate will react with the enzyme and again forms blue color like this. Now coming to the sandwich ELISA. In this ELISA we will take a polystyrene microtiter plate, and the surface of that plate is coated with antibody. Now we will add the sample containing antigen, then again add enzyme linked antibody. So antigen will be sandwiched between two antibody layers. Now we will add specified substrate which will react with the enzyme and gives blue color. Now coming to the competitive ELISA, which is used for very low concentration of antigens. In this type of ELISA, we will take antigen A and a specified amount of antibody in a separate vessel. Now this solution contains antigen antibody complex and free antibodies. After that, we will take a microtiter plate which already contains a specified amount of antigen B. Now we will add the solution to the microtiter plate, so remaining antibody will react with the antigen B which is already present in the microtiter plate. After that, washing is done, so antigen A and antibody complex will be washed out. Now enzyme linked secondary antibody will be added which will react with the primary antibody. After that, a specified substrate will be added which gives blue color. Now in next step we will add H2SO4 which leads to the appearance of yellow color. If absorbance in the colorimeter is higher, then concentration of antigen A will be lower. If absorbance in the colorimeter is lower, then concentration of antigen A is higher. For more details, please watch the video lecture of ELISA test available in our channel.
So that is all for ELISA basically. So I hope you will get it. 1 minute.
Welcome back, guys.
So this
In
is
this
also
video
one
tutorial,
video.
we'll be talking about radial immunoassay. In this radio immunoassay, ELISA can give us the idea of. So if we can detect that presence of a particular antibody, we can be sure that the infection is occurring. So that's why ELISA comes in to give us the idea what kind of antibody is present and what kind of antigen is present in our blood, in our serum. Okay. So if we see here, it actually is it's an assay, it's a technique that's called ELISA assay or ELISA technique, whatever you say. It contains particular some type of glass vessels like this one, as you can see here. This is called as the microtiter plate. This microtiter plate is nothing but plate containing small wells, as you can see here, the well-like structure. Now these wells are, there are different types of structure. Microtiter plates are available. Some of them contain 96 wells, some of them even more wells. Usually they contain 96 wells. So 96 small wells are present here all around this. So what we do now, we add the sample at the first in this particular wells. Now the thing is, this is not just a well containing slide. It has certain other very important properties too. For example, if we zoom into one of these wells, what we can see here, there might be different structure, but generally if I zoom into a well, you will look something like this. If you look from the longitudinal section, you can see this. These wells are covered with specific type of antibodies. Remember? So let's say these are the antibodies, the Y-shaped structure. They are added, they are attached to the well bottom. So at the bottom of the well, all these antibodies are attached, and their AB portions are free to react Now in this type of wells, what we can do, we can add sample now, because our sample may contain antigen. If our sample contains antigen of a particular infection, we can diagnose that particular disease. So let's add that antigen. Here the antigens are depicted with these triangular shaped molecules. So once we put the antigen in the place, those antigen will specifically interact with the antibodies that are adhered to the bottom of the wells. Once they adhere there, what we do a washing stage each time, because for the unbound antigens to be washed away.
I think I will share this video with all of you. I hope you will get the point, or you want to see this video. I think it is clear to all of you basically. So if you want, I will send it in a group. You will see this video when you will be free, or you want to see it now. Sorry for just pausing it in between, but I basically want to know that you must know the point which I want to say. So are you get my point? So kindly reply please. Because I have covered the important point which is there in ELISA. So if you want to see this video, I will later on share with you. Yes, okay. Is it okay? Is it comfortable? Because we have seen one video. So that is much for understanding. If you still want, then I will share it within a group. I save it now, so I will share it in a group. So this is basically for the, you can say this is the ELISA basically. So let me complete my presentation. So I just give me 2-3 minutes more to complete it. So this is the things which we have done. We have already know about this, that we have a antigen antibody interaction, and then we will able to know the concentration of the color change. So these are the types of interactions you have to remember. We have direct type of ELISA, not type of interactions. These are types of ELISA. We have a direct ELISA, we have indirect ELISA, we have sandwich ELISA, and then we have a competitive ELISA. 4 types of ELISA are there which you have to remember. The direct ELISA is the simple one which is just the antigen antibody interaction. Then we have indirect ELISA, in which we have antigen, then we have primary antibody, then we have a secondary antibody which is labeled with an enzyme. So this is also we have done. So this secondary antibody, which is AB2, has a specificity for the primary antibody. So this is the important thing, that the antibody 2 will definitely bind with the antibody 1 only. So this is the generally which the ELISA which we are using is the indirect ELISA. Indirect ELISA is more important. So we have indirect ELISA. So yes, they are again saying that by detecting the concentration, or by measuring them quantitatively or qualitatively, we are able to know the interaction between the antigen or antibody. So this is the indirect ELISA, in which we have antibody 1, and for this antibody 1 we have a specific antibody 2 which will be bind with the enzyme. So this is the indirect ELISA. Like here, this is the, if you are able to see here, the color is blue here, but when the enzyme is here and we are adding the substrate, there is a change in the color of the buffer. So when any this is the thing they are saying, I am not reading it thoroughly. So when antigen antibody, there is an interaction between primary antibody and the secondary antibody, then after it free antibody 2 is then washed away and substrate for the enzyme is added. So here we added the substrate. So this substrate will bind with the enzyme, and enzyme substrate will give us a particular color. So the amount of color reaction produced that form is measured by spectrophotometric plate readers, which can measure the absorptions of all the wells of 96 well plates. So we have to measure the absorption of the color. So indirect ELISA is basically used for HIV virus or HIV disease, which is a positive agent of AIDS, and even ELISA is used in your research purposes also. So in this assay, the recombinant envelope and the core proteins of HIV are absorbed. So what happened? The proteins of HIV and the recombinant, they will absorb as solid phase antigens to microtiter wells. Basically what happened, there is an interaction between the antigen and the antibodies from the HIV virus. So this is the only thing you have to remember. Individual infected with HIV, those persons who has a HIV or have AIDS, will produce serum antibodies. So these serum antibodies will bind with the antigen specifically, and there is a change in the color of the buffer solution, you can say. So individual infected with HIV will produce serum antibodies to epitopes on these viral proteins. Generally, serum antibodies to HIV can be detected by indirect ELISA within 6 weeks of infections. Because of the change in color or because of the absorption by the spectrophotometer, we are able to detect the disease. So you have to remember that ELISA is basically used for the detection of the HIV virus. Then we have non-competitive, in which we have sandwich ELISA. In sandwich ELISA, we have the antigen in between the two antibodies. That is a sandwich. That antigen is in between the two. Then we have competitive ELISA also, in which we have to add some another types of antigen antibody mixture, and then we will see the change in the color. So these are the types of antibodies. You have to remember about the indirect ELISA basically, because it is used for the detection of HIV virus. Then we have sandwich ELISA, which is basically used to know the tumor markers or hormones imbalance basically. It is used for that purpose. Then we have competitive ELISA, which is usually used to detect the antigen, because there is a competition between the antigen antibody interaction, and it is generally used for the detection of the testosterone level in our body. So these are the types of ELISA. This is again the same thing. We have antibody, we have a protein, and then we have again the second antibody, and you will see the change in the color from this one to this one. This is the main thing, only the change in the color. So I think that's all for ELISA. It's more than enough. So these are some things which they are saying, that we have to take the precautions during the washing, and washing should be these techniques takes lots of patience. So it will take 2 hours or 3 hours or 4 hours to get complete. So you have to do it very cautiously. So these are the things only. So basically it is used for the detection of HIV, like HIV-1, HIV-2, then hepatitis B, then hepatitis C and B, then another these are the applications for HIV-1, HIV-2, hepatitis C, hepatitis B. Then we have the hormone levels in our body, like HCG, which is at peak during pregnancy. Then we have luteinizing hormone, we have testosterone, we have thyroid TSH hormone, which is basically thyroid hormone. Similarly, we are able to detect some sexually transmitted disease also with the help of this amplification, with the help of this ELISA application. So these are the basically the, you can say, the techniques. So that's only for ELISA today. So are you able to get my point? Basically, what is the ELISA and what is their basic principle? Our main motive is to know the basic principle on which the technique is working. Maybe I am little bit fast today. I don't know. I want to do it fast, because we have to start the recombinant technology also. That is why I am little bit fast. So kindly let me know if is it okay to all of you. Are you able to understand ELISA? If there is your friend or someone with you, are you able to tell them what is ELISA, or are you able that the person will understand from you about ELISA? Kindly let me know. अगर आपसे कोई ELISA के बारे में पूछेगा, are you comfortable to tell them? So if you are here, then please let me know. Yes, we have Priya here, Rahul, Tanmay. Kindly let me know, Meenakshi. Yes. Clarice is also here, Vishakha. Kindly let me know. Rituja, where are you? So it's a very easy topic. I want to complete it basically on Sunday. but we are not able because we all get tired, so that is why we are not able to complete it on Sunday. I want to complete it after PCR, basically just after PCR, but it's okay, we have completed it now. So that is all for the ELISA basically. Now we have another technique, which is recombinant DNA technology. So we have should we start it today or we will continue it tomorrow? Kindly let me know, because we have only 25 minutes more. So should we start it today or we will start it tomorrow? Tomorrow we will have our full session of 2 hours. So you don't need to worry about the classes. Even we can take it much farther if you want to. And recombinant DNA technology is your subject which you have all known. Are there any short topics, ma'am? Are there any short topics? Sorry, I'm not getting your point, Devashish. Not a short topic, basically it's a part of a molecular techniques. So I'm just saying that I have taken it along with PCR, but we have done PCR yesterday, so ELISA is short for today's session. That's what I am saying. No, we can't complete it within 25 minutes, not at all. It needs full hour because we have applications of it also. So I think we will start it tomorrow. I think that's all for today. We will complete the recombinant DNA technology along with its application. So it's not for 20 to 25 minutes. It's a long topic with full discussion. So even recombinant DNA technology has 2 parts, which I have to cover it in a single part. So we will start it tomorrow if it is comfortable to all of you. We have not done the introduction of the MBBT topic 1. Okay, we will do the introduction. You need not. Yes, that's why I am asking you. If anything is left, kindly let me know. We will do it. We have ample of time with us. We have ample of days with us. So you do not need to worry. You don't need to worry. Basically, we will do the basics of or the introductory of the MBBT. No, after recombinant tech we don't have any type of another short topic we don't have. It may be of 1 hour each. If anything is left, then it may be of 1 hour, definitely not any short topic. The only thing which I know is that you have recombinant DNA technology which is left, and then now Jeet is just remind me about the introductory of the MBBT. That is the introductory session of the MBBT. So we have 2 topics right here. If any another thing is left, kindly let me know. Are you getting my point? Now my only topic which I have to do along with you basically is recombinant DNA technology, which I have to do thoroughly. So if any another topic is left, then kindly let me know. Even you can message me in our WhatsApp group also. So something if we have not done or we have not covered, then kindly let me know, because these are only the topics which I am know, or these are the only topics which basically. No, I am not taking CRISPR in recombinant DNA technology. I am starting with very basic, and I will follow all the things which are there in technology, recombinant DNA technology, but will not cover CRISPR as much, or not cover it very briefly. So just we will start recombinant DNA technology from very basics. Like every topic which we have covered, even if it's flow cytometry, even if it's ELISA, our main focus is only on the basic or the principle, so that these things will always remain in our mind. I am not taking it to the advanced level, no. I just keep it a simple one. Even if we have done blotting or SDS, you know the basic principles, that what's the difference between stacking gel or separating gel. So my focus is basically on the basic principles, so that you should not forget the basic things. That is why we have keep it very simple, very simple. I am not taking it to that much advanced level. My only priority is to know you or to tell you about the basics first. But ma'am, if you do that would be favor on us. Yes, I will try CRISPR. There is no issues in it, but I will let you know tomorrow after recombinant DNA technology, because that is the only topic which is provided to me basically, and I have planned my schedule according to it only. So there is not an issue. We will discuss CRISPR also, but kindly let me know. So I will tell you tomorrow. Within this session, I will tell you tomorrow. So there is not any worries regarding it. Yes, Priya, we will continue tomorrow. We will continue tomorrow with a new topic, with a new energy and with a fresh vibes. So tomorrow we will be there with you with recombinant DNA technology. GT is saying plant tissue culture. GT, if you have these type of topics, kindly let me know or share with me in a WhatsApp chat, so that I will ask questions and we will do it. Because right now I am not aware of these topics like CRISPR or tissue culture, but if it is there in your this session, then we will cover it. There is no issue in it. Okay, yes. So you all are very good students and you are very cooperative also. So I hope you will understand everything. There is no issue regarding anything. You are most welcome, because you are my first batch as an online interaction. I have my online interactions during my university classes also, but as a new student, you are my first new student, so you are always welcome. That is why I am asking you to keep in touch with me, and even I want you to join my next session, which is from 8 p.m. So I just want to be there also. You are my students, so I just prefer them to be there also. Okay. Devashish, I am not getting basically your point. So you can chat with me, or you can message me in your group session. So he is asking, actually Simarjit sir also did these topics, ma'am. Hence we are asking which topics. CRISPR technology, sir has covered it. Okay, I am not getting. Jeet is the day 2 topic of MBBT. Tissue culture, plant tissue culture you are asking. No issues, we will cover. Just message me in group, or even you can message me, so that I will just come to know about it, because the topics which are provided to me are only these one which I discussed with you. So if anything, okay, the plant tissue culture and CRISPR. Okay, maybe sir has lots of knowledge of CRISPR, maybe that's why they share it, or they have taken your proper lecture on CRISPR, like 1 hour lecture on CRISPR. Sir has taken 1 hour lecture on CRISPR technology. Devashish sir has taken 1 hour lecture on CRISPR technology. Kindly let me know. We will cover it. No, you don't need to worry. We will cover it definitely. There is not an issue at all. So if any other thing is there, kindly let me know. And those students who have not there on Sunday, it's my request to all of you, it's my humble request that kindly go through the previous videos and just see the things which we have done yesterday. So please go through the things. Okay, he took CRISPR technology along with recombinant technology. So you have done CRISPR already from sir. But okay, we will cover it in recombinant technology. You need not to worry. Don't worry. We will cover it. Any other thing? We have CRISPR technology left, and then we have plant tissue culture. Is it okay? Or something is left also? So we have lots of students are here. We have Rohit is here, we have Shubham, Cloris is here, Rituja, Siddhi. So kindly reply, where are all are you? You also reply something or you also say something. Is it okay? Are you getting my point? I hope you will get, definitely. And also just like my videos, I'm putting lots of effort to just doing these things. So please also like my video. Only 6 students are liking it and rest are not liking it. So kindly like it also. You are my students. Kindly like it also. Yes, they are here. So we miss those topics. Yes, ma'am. So okay. Devika is saying some students were shifted to MBBT from proteomics. Okay. Oh, no issues at all. We will cover it. No issues at all. I am saying I will take your lectures for 1 month. I have no problem in it. So we will cover it. We have 2 and 3 days more also. So we will cover it. There is no issues at all. Okay, I will check it. The topics which are given to me are that one. I will check these topics which you are mentioning, G. And I am also requesting you to write these topics and mail me, not mail me, so WhatsApp me also. So I am requesting to you, Jeet, if the topics which are left, kindly WhatsApp me. So kindly WhatsApp me the topics which are left, so that I just prepare the topics, or then I will share with you. So I think that's all. Okay, he gets my point. Okay, very good. Devashi, if you have any other query or if you have any other query or you want to ask me something, you are also welcome to message me or something like that. Even any other student. We have Rion here. So Nupur is also here. Gayatri is here. Meenakshi is here. So if you have any other query, you can message me. Sometimes we are not able to get it immediately from this chat, so you can message me also. So I think that's all for today. Kindly like the video also and kindly subscribe to the channel also, so that we will meet at 8 p.m. also with biochemistry, in which I have to cover nucleic acids today. Hope you will be there. Okay. So I thank you all for there with me, even with yes, because sometimes what happens, we are not able to get immediately the things which you are writing here, so, or even I forget also, so you can message me also. There is no issues in it. Oh, and is there any another student who is in the internship program, or is there any other student who is writing a review article? Then please kindly let me know if you are here or something like that. Floris is here and Bebita is here. We have Gayatri, we have Ankita. Lots of students, good students are present. Mitali is here. Mitali, how are you? Ma'am, I hope you go in this flow for biochem too. Yes, actually I'm quite nervous today because that will be my first lecture series for biochem, and I'm little bit nervous today. That's why I'm just closing your lecture on 6:50, so that I will get some more time to prepare for biochemistry, because within 1 hour I have another class. So I'm little bit nervous because it's the first day. You have lots of students, so first day is always I feel nervous. I don't know why I feel nervous, but I feel. Okay, so like when we were there for the first day, the very first day, when there is no connection, or we have so much problem in the connectivity issue, and at that time also I was so nervous. The first day is always nervous for me. Okay, they are your classmates. Okay. Then it's okay if they are your classmates. Because in biochem section we have students from different area. They are from your college, then they are from IIT, they are from Gujarat, they are from Rajasthan, and they are from Guwahati also. That's why I'm little bit nervous, but hoping for the best. Yes, Prati, there are some new people there. They are from different areas, from different areas of the world, of the country, they are from different states basically. That's why I am nervous. Okay, so that is all from my side today. We will meet tomorrow with recombinant DNA technology, and then we have another topics which we have to do. We will do it. Till then, you take care of yourself, and just want to say, revise all the things which we have did till now. Okay, Jeet. Yes, I will go with the speed that everyone should understand. Yes, this is a very important point which he mentions, that sometimes we speak in our own flow. Like I feel today I am little bit fast, otherwise I speak very slow. But sometimes what happens, the trainer or the teacher, he or she is in her own zone, and the students are not getting what the teacher is saying. So that is very important, to be compatible with each other or to interact with each other. Sometimes these things happen, that we are only one who is speaking, and the student is not getting any point. So this is another thing. Thank you, Ji. Thank you so much. So that's all from my side, and you don't need to say thank you to me. You all are my students, so you don't need to say thank you. It's always welcome from my side. So just take care of yourself. So from my side, a very good night to all of you, and we will meet tomorrow. And kindly go through the yesterday's session and also like my videos. Okay, so thank you all. We will meet tomorrow, or maybe in the next session, which is at 8 p.m. So bye bye to all. Bye. We have Jeet, we have Floris, we have Vaishnavi, Nupur, Manpreet, Riya. So very good night to all of you, and we will meet tomorrow. Bye. Ma'am, you are going to send the PPTs. Actually, what happens, these PPTs are there in my laptop, and WhatsApp is on phone, and there is also WhatsApp. I installed the WhatsApp on the laptop also, but I am unable to share it on WhatsApp from this laptop. So if any one of you will send me your mail ID, then it will be comfortable to me also, so that you will forward it to your friends, or you will forward it in a group, because I am unable to send it from laptop to this WhatsApp. That there is a WhatsApp here, but I am unable to send you. Even I tried today. Yes, I will mail you, so you will send me your mails. Like Juvedia is here, or your CR is here, Devash is another student. You will send me your mail. I will send you, and then you will send it in your class or in your batch. Yes. Not here in my WhatsApp group. I will forget from here. You will just send me in your mail ID in WhatsApp group. Okay. Yes. So we will meet at 8 p.m. Till then, take care of yourself. Bye to all of you. Take care of yourself. Now I am going end stream. Okay, because I have to do. So okay, bye. We will meet in the next session. Bye. Take care of yourself. and just yes, so I am also just requesting you to be there with me in the next session also, okay, that is around 8 p.m. so till then, bye. Okay, bye.